Okay, so hello everyone. So here we are again with our theoretical topics this week. We are moving on from metabolism of proteins and amino acids to metabolism of nucleotides, as I promised last week. So today I have chosen to tell you a few information about the metabolism of pyrimidine nucleotides. Since you have already had certain information about the purine metabolism when we had the exercise determination of uric acid in blood, and then I had my time to go into the metabolism of purines a little bit. So this story on the metabolism of pyrimidine nucleotides is slightly shorter, I think a little bit easier, so I'm gonna start with this and we're gonna see how we're gonna continue the topic in the next video. So regarding the fact that this is not a time-consuming topic, I'm gonna take my time informing you about the digestion of nucleotides. So what do we know about nucleoproteins? Those are heteroproteins, complex proteins, containing two parts of the molecule. Protein part of the molecule consists of homoproteins, which are predominantly, if you remember our general biochem course, those are predominantly histones or eventually protamines. And those protein moieties in nucleoproteins exhibit a slight alkaline character. The prosthetic group of those nucleoproteins are nucleic acids, DNA or RNA, deoxyribonucleic acids or ribonucleic acids, both of them with a slight acidic character. Our diet is rather rich in nucleoproteins, both of plant and animal origin. So we have to follow the digestion of nucleoproteins in two separate pathways. The first pathway is digestion of the protein moiety of nucleoproteins, and those protein moieties are actually subjected to the identical digestive pathway as we have discussed for all other protein molecules. But here I'm gonna take a couple of minutes to explain how nucleic acids themselves are digested in our gastrointestinal tract. So, digestion of nucleic acid starts in the stomach, and because of the very low pH of the stomach juice, DNA and RNA from dietary sources are denaturated and thus separated from the protein moiety and exposed, actually the structures and bonds exposed to the action of other digestive enzymes. So the digestion actually continues at the level of duodenum, as like for all other biomolecules. We have a couple of pancreatic enzymes which are active in duodenal juice, and those are pancreatic deoxyribonucleases, which are actually breaking down huge nucleic acid molecules into oligonucleotides. Then we have the action of phosphodiesterases, because polynucleotides and nucleotides in a polynucleotide are connected by phosphodiester bond. So these enzymes are actually responsible to cleave those phosphodiester bonds, thus producing 3' or 5' mononucleotides. Then we have a set of nucleotidases, which are actually breaking down nucleotides to nucleosides and removing phosphate group at the same time, so nucleosides may undergo absorption process. Also, we have a bunch of nucleosidases, which are actually responsible for cleaving nucleosides to nitrogen base and sugar moiety, which is actually phosphorylated, so to deoxyribose 1-phosphate, and those products are also absorption ready. So absorption occurs at the level of small intestine and predominantly nucleosides are absorbed. So what about the catabolism of pyrimidine nucleotides? So pyrimidine nucleotides like cytidine monophosphate, uridine monophosphate, and deoxythymidine monophosphate can be subjected to catabolic pathways, and there are several enzymes, and you can see some of them listed here and some of them are going to be listed in the next slide, are involved in such a catabolic process. So the first enzyme is nucleotides, which is actually responsible for removal of phosphate group, thus forming nucleosides. Cytidine, uridine, or thymidine. So we can take a look at this. So here we have cytidine monophosphate, uridine monophosphate, and here in brackets, if we have a methyl group, here we have deoxythymidine monophosphate. So upon the action of nucleotidases, we get cytidine, uridine, or deoxythymidine. In the next step, cytidine has to be converted to uridine in order to be subjected to further degradation by cytidine deaminase. So this is the enzyme number 2 involved in such a catabolism. So cytidine is converted or deaminated to uridine. The next enzyme is involved in transformation of uridine and the removal of sugar moiety is uridine phosphorylase here. So ribose or deoxyribose 1-phosphate are removed from our nucleosides. So we get uracil or we get thymine. So we get nitrogen bases released from nucleosides. And we are moving on to dehydrouracil dehydrogenase. So uracil and thymine are subjected to reduction process or to saturation of the double bond, and the donor of hydrogen ions are NADPH coenzymes. So here we have the enzyme listed, dehydrouracil dehydrogenase, requiring NADPH. So we have dehydrouracil and dehydrothymine forms, so they are subjected to the cleavage of the heterocyclic ring by the enzyme hydropyrimidine hydratase to form two products, beta-uridopropionate or beta-uridoisobutyrate. Those products are then subjected to one more deamination process by uridopropionase to form beta-alanine and beta-aminoisobutyrate. So let's summarize. The degradation of uracil yields the formation of beta-alanine as the final product of metabolism, and thymine, when subjected to catabolism, forms beta-aminoisobutyrate as the final degradative product. But if you take a look at those two molecules, those are amino acids, so they can be subjected to further degradation by specific aminotransferases. So transfer of amino group is accomplished by the transfer to alpha-ketoglutarate, which forms glutamate, and beta-alanine is converted to malonic semialdehyde, and beta-aminoisobutyrate is converted to its keto acid methylmalonic semialdehyde. And the last step of this degradation is activation of those two intermediates by coenzyme A to form malonyl coenzyme A and methylmalonyl coenzyme A. If you agree with me, those two final products are very well known to us because malonyl coenzyme A is an intermediate in the biosynthesis of fatty acid, if you remember, and methylmalonyl coenzyme A is an intermediate in the catabolism of fatty acid with an odd number of carbon atoms. So this is how pyrimidine nucleotides are degraded. So they can be fully degraded. Its heterocyclic ring can be cleaved and degraded to form some of the intermediates of the metabolism and finally catabolized in carbon dioxide and water. But we have to be aware of the fact that the energy which is released from those processes is like nothing, so we are actually not using the degradation of nucleotides in order to obtain energy for cell functions. So what about the synthesis of pyrimidine nucleotides? Apart from the molecule, I'm sorry for this Serbian, I forgot to translate it, but I'm gonna tell you in English, doesn't matter. So opposite to the purine nucleotides biosynthesis, in this case first we have the biosynthesis of the nitrogen base, and then the nitrogen base is attached to ribose 5-phosphate. During purine biosynthesis, it is the opposite. We are starting from the activated ribose 5-phosphate molecule, PRPP molecule, and then we are synthesizing purine ring. So the origin of the atoms in pyrimidine ring are as follows. So the first atom in the heterocycle, which is nitrogen atom, it originates from aspartate molecule. The second member of the ring, which is carbon atom, originates from carbon dioxide or bicarbonate ion, which is its soluble form. Third member of the heterocycle is nitrogen atom, and it originates from an amide group from glutamine. And fourth, 5, and the sixth members of the pyrimidine ring, those are carbon atoms, and they completely originate from aspartate molecule. So here we have an overview of the biosynthetic pathway of pyrimidine nucleotides. And if you can see, the very first reaction of the pathway is the reaction between glutamine and carbon dioxide or bicarbonate ions, employing 2 molecules of ATP to form carbamoyl phosphate. And this is the reaction: glutamine plus bicarbonate plus 2 ATPs to form carbamoyl phosphate and 2 ADPs. If you followed me carefully so far, you will recognize that this reaction is actually analogous to the first reaction of urea cycle, but with just slight differences, because in urea cycle the donor of the nitrogen atoms for carbamoyl phosphate was ammonia or ammonium ion, and here we have glutamine as a molecule which can fix ammonia in its amide group in a side chain. And the second one is that this reaction in pyrimidine nucleotides biosynthesis occurs in cytosol, and this reaction in urea cycle occurs in mitochondria, if you remember. The enzyme which catalyzes this reaction is called carbamoyl phosphate synthase or synthetase, and it is the same enzyme but different isoenzyme. So this one is marked with number 2, so carbamoyl phosphate synthetase 2, which is located in cytosol and not in mitochondria like its isoenzyme form, carbamoyl phosphate synthetase 1. But regardless the type of the isoenzyme, generally speaking, carbamoyl phosphate synthetase enzymes are regulatory enzymes. So like in the urea cycle, here as well, this reaction is regulatory reaction, and CPS2 is regulatory enzyme like it was CP1 in urea cycle. In the reaction number 2, we have the condensation or the transfer of the whole molecule of aspartate to carbamoyl phosphate to form carbamoyl aspartate. Despite the fact that we have the formation of the new chemical bond, ATP is not required for this step, because we have to take into account that carbamoyl phosphate molecule is already activated molecule. And the enzyme catalyzing I'm sorry, this reaction is aspartate carbamoylase. The third reaction is the reaction of cyclization, and this step actually is completed in two steps. The first step is catalyzed by enzyme dehydroorotate, and dehydroorotate is formed, and then there is an oxidation going on by dehydroorotate dehydrogenase, and this is the enzyme which is only found in mitochondria. The rest of the enzymes are located in cytosol, and it requires NAD plus as acceptor of reducing equivalent. So if you take a look here, the final product of those two steps in the reaction number 3 is actually the formation of orotate or orotic acid, which is actually the first pyrimidine base synthesized during this pathway. Now, when we have the first pyrimidine base synthesized, we have to attach sugar moiety. So in the step number 4 or reaction number 4, we have the binding of ribose molecule, ribose 5-phosphate to orotic acid or orotate, and the reaction is catalyzed by orotate phosphoribosyl transferase, and the donor of ribose group is of course activated phosphoribosyl pyrophosphate. This enzyme is also active in the salvage pathway of pyrimidine nucleotide synthesis, and the defect of this enzyme is actually the basis of development of hereditary orotic aciduria, which arises as a consequence of accumulation of orotic acid in the organism. So oritidine 5-phosphate or oritidine monophosphate is the first pyrimidine nucleotide to be synthesized during this pathway. So from OMP we can now synthesize rest of the pyrimidine nucleotides, and the first in a sequence is uridine monophosphate. Uridine monophosphate is synthesized from OMP by decarboxylation reaction. So carboxylic group is removed in the form of carbon dioxide to form uridine monophosphate. The reaction is catalyzed by orotidine 5-phosphate decarboxylase. Let me make just a small digression. In mammals, first 3 enzymes of the pathway, so carbamoyl phosphate synthetase 2, aspartate transcarbamylase, and dehydroorotase, are located on the same protein carrier, and this complex is marked as CAD, upcoming from the first letters of the name of these 3 enzymes. And the last 2 enzymes are also located on the same protein carrier, and they are marked by the name uridine monophosphate synthase or synthetase. So after uridine monophosphate is synthesized, we have to phosphorylate it to UTP. So we have UMP, and phosphate groups are donated from ATP molecule to form UTP. And then from uridine 3-phosphate we can synthesize titidine 3-phosphate, and actually the donor of the amino group for attachment to the carbon atom number 4 of pyrimidine ring is actually glutamine, and its amide group down there. So the enzyme which catalyzes this reaction is CTP synthetase. And of course we have now we have UTP, we have CTP, so they are precursors for the RNA synthesis, and they can be incorporated in different types of RNA molecules. What about the regulation of the pyrimidine biosynthesis? So we have already mentioned that we have one major regulatory enzyme, and that is carbamoyl phosphate synthetase, and this enzyme is inhibited by UTP and CTP and activated by ATP. The activation by ATP is actually accomplished through the formation of phosphoribosyl pyrophosphate, And the second site or second point of regulation may be OMP decarboxylase, which is inhibited by both UMP and CMP. But inactivation of OMP decarboxylase actually cause the inactivation of orotate phosphoribosyl transferase 2, and this enzyme as well becomes dysfunctional because, if you remember, they are located on the same protein carrier. So that is the reason why the defect of this enzyme ends up in the accumulation of orotate or orotic acid and not OMP. What is the salvage pathway of pyrimidine biosynthesis? Recyclization of pyrimidines is less important than for the recyclization and reutilization for purines, which is of ultimate importance. So why is that so? Because pyrimidines are degraded, if you remember from our previous slides, to soluble metabolites, beta-alanine or beta-amino isobutyrate, so they can be excreted from our organism, ending up in the fact that the concentration of those metabolites in blood is rather low, so the intensity of recyclization is also gonna be at low levels. But it's still possible, and pyrimidines can be converted to nucleotides, and the enzyme is pyrimidine phosphoribosyl transferase. So any of the pyrimidines, the orotate, uracil, or thymine, can undergo reaction with PRPP to form pyrimidine nucleoside monophosphate. But cytosine cannot undergo this salvage pathway. When there is a need to synthesize deoxyribonucleotides for incorporation in DNA molecule, we need to reduce sugar moiety, so we have to remove OH group from ribose to form deoxyribose, and the enzyme which catalyzes this reduction is ribonucleotide reductase, and this is the only one for all of the deoxyribonucleotides, and it requires, because of the active site and the main point site for reaction are actually SH group, which are donors of hydrogens for this reduction, and it requires a protein coenzyme called thioredoxine, which has those SH group which can be converted to SS bridges, as we mentioned a couple of times in previous chapters. So what about Dttp, which has to be incorporated in the molecule of DNA during DNA synthesis de novo? And if you remember the basics from general biochemistry, thymine is nothing else but methyl uracil. So the major reaction for the biosynthesis of DTTP is methylation of deoxyuridine monophosphate to deoxythymidine monophosphate. And if you remember from our previous videos, whenever we have a methylation, we have repeated it so many times, we require tetrahydrofolate, in this case N5N10 methylene tetrahydrofolate, and the enzyme which requires this coenzyme for methylation is thymidylate synthetase. During this reaction, methylene tetrahydrofolate is converted to dihydrofolate, so apart from methylation, THF also donates 2 hydrogen for the reduction. So there are some molecules which are inhibitors of this enzyme thymidylate synthase, and those compounds are actually found to be rather good, if we can say that they are good and effective cytostatics, like 5-fluorouracil or 5-fluorodeoxyuridine. So if you take a look at this story, you will notice that THF is used up to form DHF. So in order to have it for the novel biosynthesis of DTTP, we have to regenerate THF. And this is accomplished by the enzyme DHF reductase. And if this enzyme is inhibited by certain compounds, they can also act as potential cytostatic molecules and compounds, because if you inhibit DHF reductase, you will inhibit the biosynthesis of, indirectly inhibit the biosynthesis of DTTP, which can be very important to prevent the DNA synthesis of fast proliferating malignant cells. Some of the compounds which are actually targeting DHF reductase are very well known: methotrexate or aminopterin or 3-methoprim, and so on and so on. So once when DTMP is synthesized, a few phosphorylations are going on, so DTMP is phosphorylated to DTTP. So if you notice here, we have DUMP converted to DTMP, and then we have phosphorylation to DTTP, which means that low levels of deoxyuridine triphosphate are maintained in a cell, which is very important because those small concentrations are actually preventing the incorporation of uracil in the molecule of DNA. And regarding at the very end of this story, the salvage pathway of DTTP synthesis requires the action of two enzymes, thymine phosphorylase, which actually binds deoxyribose 1-phosphate moiety to thymine, forming nucleoside thymidine, and thymidine is further phosphorylated by thymidine kinase to form DTMP, DTDP, and DTTP, ready for incorporation in DNA molecules. So more or less that was it regarding pyrimidine metabolism. Again, I'm suggesting you that you can follow the purine nucleotide metabolism by following our exercise on uric acid determination in blood, but until some other time, and probably the story on purine metabolism, stay healthy, stay safe, and bye bye.